Good morning. I'm Gula Lipschitz, and it's now my pleasure to introduce the interim CEO of Century Therapeutics, Dr. Greg Rosati. Thank you, Greg, for joining us. The format for this session is about 25 minutes fireside chat, but we'll start off with a few high-level overview slides of Century and its manufacturing capabilities before we move on to the Q&A portion. If anyone has any questions during the session, feel free to type them into the question box and we'll try to ask them on your behalf. Greg, thanks again for joining us. Can you start off by giving us an overview of Century and its manufacturing process, and then we'll dive into the details a little bit more.
Great. Thanks, Gula. Thank you for the invite. Thanks for having me here today. Excited to talk about Century and particularly our manufacturing approach and process. So I'm just going to give a quick overview with a few slides, and then happy to go back to these slides to answer the questions. So just to start off, Century has many assets and platforms that are important to our success. These include the ability to reprogram cells into iPSCs, and we have quite a collection of clinical grade lines. We have a very dedicated, talented protein engineering group that knows how to make next generation CARs and many other things. We can put those CARs along with other edits on our cells using very precise CRISPR editing using the MAD7 protein. And we have differentiation protocols towards NK and gamma delta T cells, as well as scalable processes and a very robust process and analytical development approach to have strong process and assays. Just real briefly here, the ability to go from an iPSC to a differentiated cell is very powerful because we can do all of our edits on the iPSC. So we do them once up front. We then get a clone that has all the right edits, things that are knocked out properly, things that are knocked in properly. And then we create a master cell bank from that clone. And from there, we differentiate to our product candidates batch by batch and very large batches. So the beauty of this is we can do very precise edits, put on what we want to put on, select the clone that has everything we want. We can manage genome integrity, and then that master cell bank allows us to have a very scalable, consistent process. This next slide shows it a little bit better in contrast to autologous and primary derived allogeneic cells. But you have to go back to a starting material every time. Obviously, with autologous, you got to go back to the patient each time is a new patient, but even with donor-derived, you have to have multiple donors to satisfy the patient needs. You have to do the gene editing multiple times. With our approach, as shown on the bottom panel of the slide, we do our editing once, create that master cell bank. So that really limits the regulatory constraints on the gene editing components as far as having to show that they're consistent, because you only make them once, you only use them once. And then the cloning allows us to avoid variability you might see from the gene editing that you might do from one donor to the next, and reduce the starting material variability as well. Genome editing, I'm sure we'll talk about more later, but and I won't go through this whole slide now. We need to think about what the agency's asking as far as how to prove that your editing is doing what you intended to do. We have multiple methods to do this, and then we have the thought process behind it is what we're trying to prove in terms of safety of the product after such edits. As you've probably heard from before from Century, in-house manufacturing is something we've dedicated ourselves to. We have our own plant in Branchburg, New Jersey, about an hour and 15 minutes from Philadelphia, from our headquarters. And we've used the partner Fujifilm Cellular Dynamics in Madison, Wisconsin for our first product and potentially other NK products. And we just feel this gives us a great advantage, which I'll talk more about later. But we feel it's central to our success. We made this very conscious choice rather than using CDMOs. And one thing I kind of alluded to earlier in the slides is the ability to scale up. And with iPSC-derived cells, there's really no limit to how much you can scale up, because the iPSC cells have unlimited proliferation potential, unlike primary donor cells, which have a limit. So with the primary donor cell, you can scale up, and an allogeneic, you can do multiple doses from one donor, but there's a limit on how much a donor can provide. Here, there's no limit. So we think we can scale much, much further and drive the cost of goods much, much lower than primary derived allogeneic. But to do that, we need expertise and investment in scale up, which we believe we can leverage knowledge from other fields like biologics and vaccines and other cell therapy spaces where there's been some success scaling up, which I've been personally involved in the past. And we recognize it's not just the cell expansion that has to be scaled up with the harvest and the final products filling as well. And with a good engineering approach and the right kind of talent, we know this can be done. We're already on a good path to getting there. So I think that's all I have for slides. Happy to go back to these again to elaborate on some questions. So yeah, thanks for listening.
Great. So maybe to start off, we'll start with a little bit of a high-level question. Can you expand a little bit on why Century decided to build out that internal manufacturing facility rather than working primarily with CDMO partners?
Yeah, sure. And we made that decision really early on. From the time I joined in early 2020 and even in 2019, as we talked about the strategy, as I was talking about coming to Century. From my past experience, I've lived this. Having your own facility means everybody from development, research, manufacturing, quality, everybody has the same goals, the same mission, the same priorities, the same interests, the same motivation. So really all working together as one team allows you to just do things faster and better. And you can see on this slide a few other points too about protecting know-how. Whenever you go to a CDMO or partner, you have the potential to expose some of your know-how to other competitors. But most importantly, like I said, it's about better and faster iteration. So we do believe there'll be better success because everyone is right there together trying to make this work. You have to troubleshoot problems, make process changes, you can be more nimble. And of course, the simple thing like control the schedule. You don't have to worry about reserving a slot. What if my schedule gets delayed? Do I still have a slot? Do I have to pay extra because now I delayed it? What if I want to go earlier? Well, there's no slot for me at the CDMO. Your own facility, you can do whatever you want. And I didn't mention this in this slide other than to say it's a 53,000 square foot facility. But our facility is a multi-suite, multi-product type facility. And with allogeneic also, it's unlike autologous where, autologous, again, I've lived this in previous experiences. Autologous, when you're running a trial, your suite is taken up by that product. And maybe you can share products, maybe not. There's debate about that. But your suite is taken up the entire trial because every patient has to require his or her own batch. With allogeneic, you campaign things. You do things ahead. You make your batch or two or three or whatever you want to make. Done with that suite, move something else in. But we do have multiple suites, so we feel like we have a lot of flexibility. Again, if things speed up, we can go in sooner. If there's delays, but most importantly, like I said, having all the experts together with the same priorities, the same motivation, we can get things done much more effectively than relying on a partner. Even though the partners have talent, they're just not as experienced with your product and not as vested in the interest of your product. So we think this is going to be a real differentiator for us. We feel it already is.
Mm-hmm. Got it. And you alluded to some cost advantages earlier. Obviously, in the current financing climate, there's considerable attention on cost. What can you share with us regarding, expected COGS for your programs or how costs might be different from autologous and donor-derived cells?
Right. And I'll just maybe toggle through a few slides. I don't have a slide on this per se, but like I talked about earlier, we can scale these things up. So when you think about autologous, the worst case scenario for cost, every patient requires a batch. Every batch requires release of that product. So all the testing you do is just for that one product for that one patient. The batch itself, all the labor on that batch is just for that product for that one patient. When you scale up, you spread that cost. This is obvious. Obviously, everybody knows this, but just want to emphasize the point. And the more you scale up, the more doses you can make per batch, the more you spread that cost among more doses. Not just the product or processing costs, but the release testing costs, the labor costs, all of it gets spread out. So we do feel we can scale quite a bit here. And when you think about numbers, I'm not going to share an exact number with you today, but I can tell you that we all heard the numbers on autologous, on the order of hundreds of thousands, 100,000. I've heard different numbers, and you can believe what you want to believe, but you can probably, anybody that understands the manufacturing can do the math for themselves and realize it gets very expensive for these autologous processes. We believe will be order of magnitude better, and not order of magnitude better of where they are now. We will be order of magnitude better of where the autologous can go. Primary derived is a little trickier because I think primary derived, I don't think anybody really knows the answer to how much you can expand those cells and still get efficacy. I don't know exactly, and I don't think anybody knows other than themselves what the primary donor-derived allogeneics are doing as far as how far they're pushing those cells for the trials, right? For me, I'd say, on the one hand, I want to be conservative and not expand them too much because I want to have efficacy. On the other hand, I want to expand them a lot because I want to have good cost of goods. Where they are today, where they're going to be later, I don't know. But there is going to be a limit. We do know primary-derived T cells, primary-derived NK cells, you can't grow them out so much because they will, especially T cells, they're going to lose their activity. They'll be anergic if you push them too far. So our advantage, like I said before, is that we're going to grow our iPSCs out to very large numbers and then differentiate batch by batch. So we don't have that limitation. We believe, again, our batches can be much, much larger, which will drive the cost of goods lower than primary derived allogeneic. Cool. For some reason, I can't hear you now. You're on mute.
Yeah. Left myself on mute. So yeah, beyond cost and expandability, are there other differences from autologous and donor-derived manufacturing that you think people might not yet appreciate as the space continues to mature?
Yeah, I think there's a couple, and we talked about it, but I think you have to really get into the details behind the process to appreciate it. So I'll just go through a little more detail on this slide, if you don't mind. So let's look at the top panel for a second, and the simple one is autologous, but even primary donor-derived. You start with a healthy donor, collect their cells, isolate what you want to isolate. Let's use a T-cell process. Isolate the T-cells or the PBMCs. You do your editing, you knock out whatever you want to knock out, HLA, TCR, etc. You knock in your CAR, whatever else you want to knock in. And by the way, here's one advantage. There's a limit of how much you can knock in and knock out, because every time you do an edit, you have to sort and grow. And again, you're burning the proliferative potential of those cells. So there'll be a limit of how many edits. So that's one advantage. There's only so many edits you can do with donor-derived. And of course, autologous is the same problem, maybe worse. The second thing is, now you go ahead and you take those primary T cells, which have been edited, and now that's your drug substance. But there's only so many doses you can get from that donor. Like I said, how far can you push those cells? So now you got to go back and you got to get another donor, do editing again. So here's problem number 2. Is that donor the same as the first donor? And you have to screen a bunch of donors to get a donor just as good. So donor to donor variability. And here's problem number 3, which is the editing is going to be variable. And from one donor to the next, from one process to the next. So your editing results, which means your quality of your final product, is going to change from donor processing to donor processing. Okay, so you have limited number of edits, you have editing variability, and you have donor variability. iPSC-derived eliminates all of that because of the unlimited proliferative potential of the iPSCs. When we get that final, can you see my cursor if I point? Can you see a cursor? Yeah. So when you get to this master cell bank, you've already done all your editing. Again, we can do more edits than donor-derived. You've selected a clone that has the right attributes, so quality. We have something that we feel strongly about, that it has the right edits and genome integrity is preserved. And we're not doing it again because this master cell bank has the proliferative potential. These cells have the proliferative potential that we can make a master cell bank as large as we want, and we can expand it before we start to differentiate as much as we want. So this master cell bank will last the lifetime of the product. And then, like I said, we don't have to go back, and our products will be much more consistent because they're all derived from the same master cell bank, which is derived from the same donor. So there's multiple advantages besides cost, and probably even greater advantage than cost, because ultimately it's about getting good outcomes for patients in terms of safety and efficacy. So we have much better control of what our product can be, and therefore we believe we'll have much better outcomes because of the ability to control these things.
Got it. So now that Century has gone through the IND process and is in the clinic with its first iPSC-derived CAR-NK, can you share a few thoughts regarding what you've learned from that process? For example, what the FDA is looking for in terms of CMC for this type of product?
Yeah, absolutely. We're really, really excited that we have product in the clinic and that we've been through the IND process. And I just want to say it's been a great collaboration with the FDA. As everyone knows, you have pre-IND meetings, but people probably also know about this Interact forum. And I forget what Interact stands for, but it's about interacting with the FDA. It's a cute acronym. It replaces the approach people used to take with pre-INDs, which were these informal meetings. But it's a little more formal, but it's still non-binding advice. And we've leveraged both of those and had great interactions as we thought about testing approaches, et cetera. And this is a new field. iPSCs, there are not many iPSC-derived products in the clinic. So it was great to learn with the FDA. They learned from us, we learned from them. A couple of things I'll just highlight, which I think you probably want to talk about, is the effects of the editing. So any clonal product, I think they're going to ask to do whole genome sequencing to really understand what you did. So we have a really good sense of approach now, because we had an approach proposed in our IND as far as what whole genome sequencing means. And I'm not an expert on whole genome sequencing, so I'm not going to be able to get deep into the science on this. But when it comes to whole genome sequencing, there's lots of different methods you can use, and there's lots of different layers of how deep you can go. You can look at really large segments or smaller segments of base pairs. So we had an approach that we thought was rigorous. Maybe I'll just show the slide real quick. We thought it was important to have orthogonal methods and also test our product regularly, not just once. Also test the master cell bank and things in between. And on the left here are the things you look for. I won't read the slide. You can read it yourself. I'm happy to elaborate if you like. But back to your question, Gula, the real great situation Century is now in is that we've been through the IND process. We've gotten the safe to proceed. We're in the clinic. We know what the FDA is looking for as far as gene editing analysis or analysis of our gene editing and genome integrity, genome stability. We're comfortable with those assays. We have a lot of, I'll be honest, we have a lot of rigor here that we've done a lot of assays and put a lot of investment in that. But we have a playbook now that we believe we can use for all of our programs. So we feel really excited. This positions us really, really well.
Great. So beyond genome editing and genome integrity characterization, can you elaborate on some of the other analytics and quality control steps that you think are important to the consistency and quality of your products?
Yeah, I'm just going to put a slide. It's always nice to have a slide to talk to. I don't know if this one says much, but when I think about the final product, it's just like any cell therapy. You need to look at safety, potency, purity, identity, of course. So it's pretty standard, but it's pretty rigorous as well. So you have safety, of course, sterility, mycoplasma, endotoxin, viral adverse agents. We do have animal source components for various steps, including the gene editing steps. So we have to make sure we put together a good risk analysis on what those different animal source components bring as far as risk. So we think about how to manage that risk by either testing raw materials or testing certain aspects. Obviously, the master cell bank, if anybody reads the guidance, they'll understand the master cell bank has to be tested very, very rigorously for those kinds of things. So that's safety. That's pretty straightforward. I can answer more questions about that if you like. And then the other things which are somewhat straightforward, purity and identity. Obviously, if you're making an NK cell, you're looking for certain markers. There are going to be certain impurities in there. Just natural when you do a differentiation process, you're not going to get 100% of your cells exactly what you want. So it's important to know what else is in there and what safety risks they bring. And we have certain parameters, obviously, like with an NK cell, we want to make sure we're below a certain level of CD3 positive cells. We don't want to have T cells in there and vice versa. And then other purity, like gene editing components, we want to make sure are no longer there, for example. And then, but the big one is potency, right? That's the big one. And we're all learning. Even the simplest case, and it's not that simple, of autologous CAR T. I think everybody knows what they're trying to do, but exactly how the CAR interacts with the antigen and what then happens in a cascade of effects, everyone's still learning. So, you hear about cytokine secretion assays, for example, interferon gamma. I think that's what the autologous players all put forward for their first BLAs, and maybe are still doing. I'm not aware if they made changes, but that's a good surrogate of cell killing, which is pretty straightforward as far as an assay that you can then validate. So here's the balance. You want an assay that you can validate because you want to have confidence that assays run the same way and you're measuring the same thing each time, but you need a meaningful assay. So on the other extreme, one could propose, well, I just want to show expression of CAR. If I have expression of CAR, that must be a potent cell. Well, that's probably not going to fly because it's just too simple. You do want to look at expression of your CAR. That's another assay we do have. When it comes to identity, you look for certain things that are expressed at certain levels. But then there's the in-between. So like cytotoxicity is the ultimate goal here, and the cascade of other effects that happen in vivo. Those assays can get really complex. So we're doing them in phase 1 as characterization methods. And our philosophy has always been lots of rigor, lots of assays, lots of understanding, but don't make it overly complicated for your release assays that you propose for your QC release for the FDA. So propose a simpler assay, but have these others that are characterization, share them with the FDA. It's always good to have a dialogue with the FDA, let them know what you're thinking, because as you learn more, it may turn out that one of those other assays elevate to the potency assay. So you want to constantly learn, share data with the FDA. So my advice to people is never be afraid to share your data, good or bad, with the FDA, because it's a learning experience. And if it's really bad and you can't go forward, that's life. What are you going to do? But most of the time it's learning. You say, oh, this is what makes the cell work. This one's less important. And then back to robustness. If this assay just can't be robust, how do you validate it? So it's the balance of all those things. Hopefully that answers your question. I know I spoke about a lot of different things.
No, I think that was actually great, and touched on a lot of important topics there. So Obviously the program that's in the clinic now is NK cells, but you also have the iPSC-derived gamma delta T cell programs that are in the pipeline. So can you talk about how similar this is to the NK cell production with respect to the equipment that you use, the components, and the read across? You mentioned that on the genome integrity side, you can use a lot of learnings from the FDA interaction, but what about some of the other elements?
Well, I'll just talk about forward-looking stuff. So I think, I know we talked publicly about our 101 product, the one that's in the clinic, which is the INK product. That one we made with static culture. So we use G-Rexes, and we've had this on some posters that our team has presented. And that was just a quick way to get into the clinic. We want to move to stirred tank or some sort of dynamic bioreactor. So we're looking at stirred tanks, we're looking at Zori wave bioreactors, and you'll see data on these in the near future at presentations. Our group will continue to publish. But yeah, to answer your question, the good news is the data we have so far, and we have looked ahead on NK for future programs, and even the 101 program, as we get further into clinical trials, we want to make a switch to a more dynamic culture so that we can have better cost of goods. The G-Rexes are nice, and you can scale them out better than you can scale out an autologous, but you can't scale them up. I think 5 liters is the biggest G-Rex, and I'm sure it's a very good technology. There probably are other ways to scale up, but it is a scale out. So stirred tank bioreactors, Zori bioreactors, which are like the old wave, the GE wave, which is now the Cytiva Zori, are ways to scale up. So we do have success right now in some dynamic cultures like that for both NK and T. So very similar. How you do it, the differentiation is obviously very different. The cytokines you use are different. Other stimulants, how you stimulate certain ligands or antibodies are different. I'm not going to get into the specifics, that's very proprietary. But the overall technologies that the process development groups use can be very similar. And then cell harvest, you see here a KSEP, which is a counterflow continuous centrifugation approach. That's something that we are moving towards as well. And then drug product filling is another challenge, which we're still early on. We do fill hundreds of vials now, not with an automated filling system like that. That's for down the road. We're going to fill thousands. We're not there yet.
Got it. So you touched on a couple of approaches and things that you're looking at to move your manufacturing into kind of the next level. Are there any other elements that you think are going to be important that you guys are looking to incorporate, for example, automation or any other technologies that could be important towards increasing either the scale or the efficiency or some other elements of your process?
Yeah. Right. So I think the beauty of these dynamic systems is that they are naturally automated. So when you think about, so I think about automation, everyone's got those hot topics of automation and robotics. I think that people are mostly thinking about how do you get labor out of these autologous systems. So the autologous systems are very hands-on. Every day you got to take a sample and measure, or maybe not every day. I think a lot of people do every day, but it's a lot of manipulations. The beauty of a bioreactor is it goes back to the biologics industry, which has been around for 30 years. People have been doing antibodies, 30, something like that, maybe 30, 40 years. So these are naturally automated systems, very low labor needs. There's feedback control. So the simple things, oxygen, pH, temperature, those are all built in. I think we're all looking at more sophisticated things, and there's lots of small companies and also academic institutions. I don't know if anybody knows the Center for Manufacturing Technologies, which is an industry academic consortium led by Georgia Tech, which we're a member of. They have a lot of very interesting technologies for online measurements. And if you could use these online measurements, simple things, let's say like glucose and lactate, but maybe more complicated things like other metabolites, where you can then use feedback loops to say, oh, now I know how the cells are going this direction. Let's ramp this up. Let's ramp that down. So the beauty again of these types of systems, these dynamic culture systems, is it's all amenable to this kind of automation. And most of it, a lot of it's the simple stuff built in already, but we are looking at the more sophisticated things. And we're very connected to these cutting edge technologies, like I said, through these consortium. There's another one called NIMBLE, which is the National Institute for Innovation and Manufacturing of Biologic or Biopharmaceuticals, I think it is. But that's a consortium that touches upon both biologics and cell therapy. So we're also a member of that one. So lots of great stuff out there. And right now we're keeping it simple because we can. But as we go further along and we learn more, there's lots of opportunities to make it even more sophisticated with more automation to have even better control of your final product quality, and also this. But the scale is limitless because the cells can only get to whatever they can get to. But let's just say, I don't know, can we grow these cells at 1,000 scale someday? I think so. The data we have so far says yes. And back to donor, I hate to disparage another area because I think there's lots of value in donor derived as well. But they can only make a batch as large as the number of doses that comes from one donor because you're not going to combine donors, right? So I don't know what that number is, but it's not 1,000 liters. I can guarantee you that. So we do have that advantage.
Great. So I see we're just about at the end of our session time. So I'd like to thank you again for Greg for your participation and for going through the Century capabilities and manufacturing approach.
No, my pleasure. Thanks for the great questions and the great chance to talk about these advantages of our platform and our company.